Good morning, and welcome to the PAG webinar series supported by Sequel. Thank you for taking the time out of your day to join us, and thank you, Sequel, for your partnership. Today's topic is the role of population scale, low-pass, whole genome sequencing in accelerating crop improvement. The question and answer portion of the webinar will take place at the end of the presentation. You can submit questions via the Q&A function at any time throughout the event. Any questions not addressed live will be answered via email. We would love to hear your feedback, so please submit your questions and comments. I would now like to introduce our speaker, Dr. Josh Clevenger, faculty investigator, HudsonAlpha Institute for Biotechnology. Dr. Clevenger is a passionate plant scientist using genomics to develop improved crop varieties around the globe. During his PhD, he played a crucial role in sequencing the peanut genome, a major breakthrough for the peanut industry. After a short USDA NIFA postdoctoral fellowship, Dr. Clevenger worked for Mars Wrigley on the Global Nut Science team, designing and deploying genetic improvement projects across 5 continents where peanuts are grown. Currently, Dr. Clevenger is focused on developing new tools to make crop breeding faster and more affordable for breeders and farmers. His work is helping to create crop varieties that are more resistant to disease and can thrive in challenging environments. I would now like to turn the webinar over to Josh to begin the presentation. Take it away, Josh.
Thank you, Drew. That was an awesome introduction. I really appreciate it. And for all of you that came here today to listen to us, we appreciate you and appreciate you spending your time with us. And I think you're going to enjoy the webinar that we prepared for you today. And before I get started, I always like to acknowledge the really talented folks that I get to work with as part of the Clevenger Lab. And those folks are on the screen in front of you. And all of them do different things in the lab to really help accelerate crop improvement, which is what we're extremely passionate about. We believe that everyone has a right to safe and nutritious food, and the things that we do every day are working towards that. And one of the things I think for you young people, or maybe you older people that haven't experienced this yet, that I think is like the moment you know that you've made it, is when your lab makes logos for you that you've never seen, and they don't even ask your permission before they put them in slides. And honestly, I love that. That is one of the most exciting things I think that I've done in my career. So before I get started with talking about the topic, I think it's important for you to understand, why should you listen to what I'm saying? What particular expertise do I have that might lend wisdom to the things that you're trying to do in your programs in your neck of the woods? Well, for the past probably 5 years here at HudsonAlpha, we've sequenced and analyzed whole genomes at scale. And we've done that across a wide variety of plant and animal species. And we've sequenced from DNA from sheds of skin of snakes, or clippings of turtles, or little shavings of seeds from all types of different plants, stems and leaves, the blood of pigs, soil microbiome. Recently, we sequenced the microbiome from the air, so DNA that the Air Force captured and extracted from the air, and we sequenced that DNA. And mostly what we do is we help folks to do the things that they're trying to do in their program. So discover those genes that are going to be impactful for the crops that they're most interested in, to help feed the folks that have stakeholders in those crops, to develop marker systems and markers to help accelerate their improvement of their plants, or sometimes just to understand the diversity of the species that they're working on. And all of these sort of things that we help folks with have a major impact in what they're trying to do in their neck of the woods. And through those 5 years and all of those tens and now hundreds of thousands of different samples that we've worked on, we've really learned a lot about what it takes to make an impact quickly. And so that's really what the sort of webinar is about today, is about when you have your problem that you're interested in, that you are passionate about, that you need to solve quickly because somebody somewhere is suffering because there's a disease, or there's malnutrition, or there's drought, or there's other stresses that you need to help solve, you want to do that as quickly as possible. And so I'm really going to take you through sort of an option of how you might do that today. And before we get started, I have to acknowledge that most of the ideas that we've been developing and working on for the past 6 years have really stemmed from conversations with friends at Calientitos in Athens, which is a Cuban restaurant. And every Wednesday, Justin Vaughn and Waleed Karani and I sat down and had tacos and talked about our ideas about how we sort of accelerate genomics and crop improvement. And what's really exciting about me when I was putting this webinar together is that everything that's in this presentation is things that we talked about at those lunches. So I think it's an important sort of life lesson to never sort of minimize the impact of good friends and good conversation and good food on what you can do in your career. Okay. So usually what I do is I sort of speak at you with things that I've done, right? I'm showing you this is what we've done with the tools that we have and that we've developed. But today what I want you to do, no matter who you are or what you're working on or what part of your career you're in, there is one thing maybe above all others that you are most focused on trying to solve. There is a trait, it might be the thorns on a blackberry, which was recently sort of discovered how to control that. It might be a disease that's really devastating for the crop that you're interested in. It might be fortifying some nutritious aspect of the plant that you're trying to work on. And so whatever that is, during this next hour, I want you to keep hold of that in your mind as we go through sort of this decision-making process of how do I most quickly get to the impact that I want to have. And so that's sort of the focus of what we're going to do today. And I'm going to take you through a story in our neck of the woods, and I'm going to show you how we moved through that story and sort of got to a good conclusion. And so this is a disease. This is peanut smut disease. And it's very scary because basically if you care about peanuts, and some of you might not, but just again, imagine the things that you do care about, and you grow that crop and it takes 140 days. And at the end of the year, when you dig up those peanuts and you open them up and they just become spores in the wind, and you have 50% of the yield, and the other 50%, no one wants to buy from you because they don't want to spread that disease across the globe. And so peanut smut actually started in Argentina about 30 years ago, gained ground in that country. And so what's really interesting about Argentina is that they grow about the same amount of peanuts that we do in the United States, but 100% of them are exported. And so this is the seriously global export market. And having this disease there limited their ability to sell their product. And so a huge amount of the country's income all of a sudden was threatened. But not only that, but smut disease itself, again, like everything in agriculture that we have to work on, the spores are incredibly resilient. They can withstand high temperatures and high and low pHs, low temperatures. They can stick to all sorts of surfaces and go all around the world. And so it really has this ability to spread. And so when the disease was really getting bad about 10 years ago, it was clear that because most of the peanuts grown around the world were susceptible to this disease, this was going to be a major global crisis for peanut, for all the things that are made with peanuts, even in commercial products, or those folks that are growing peanuts for their own consumption all around the world. So we really needed to have an extremely good tool to combat peanut smut resistance. Okay, so you're a researcher, I'm a researcher. We have a problem, okay? We decided that we're gonna work on peanut smut. Maybe we got some money from our government to put a project together. Maybe we're being paid by the growers associations. What's the first thing we might do? Well, the first thing is you're gonna wanna see if you have a resistant line that you can use. And in this case, we tested over a couple years and found some germplasm that looked to be resistant. So that's wonderful. But of course, what I've found in agriculture is that every time we have this incredibly impactful trait, especially in disease resistance, it's within a line that no one probably is going to grow commercially. It either matures too late, or it doesn't taste good, or the grain is not the right size and shape, or the yield is poor. And so we really have to bring that trait into a agronomically elite background. And what I'm showing on the left of the screen there is a population that was made from crossing a resistant peanut to a susceptible peanut, and those are F2 plants. The other thing that we always deal with in these situations is that, are we in an inbred crop, are we taking our populations to fixation? Meaning, are we selfing those populations to an F6 or an F8 stage where all the DNA is presumably fixed in those lines and we have pure lines to do the genetics on? In this case, that would have taken us another 2 or 3 years before we started to do the genetic mapping, and we really didn't want to take that much time. So I'm actually showing you the population in the field. It was about 600 plants where we were able to look at phenotyping those plants and trying to have a strategy to actually understand the genetic mechanism for what was controlling that smut resistance. Now, I have to tell you about the phenotyping for this because it's incredible, okay? So you have a soil-borne fungus that infects the grain underneath the soil, and some of the seeds it infects and some of the seeds it doesn't infect. So if you're putting on your sort of phenotyping hat, because in genetics, which is kind of interesting, is that the phenotypes are way more important than genotypes in some situations. So if you put on your phenotypic hat and how you might do that, the best way to do it is to take every one of those individual plants and harvest all the seeds and lay out very carefully all of those seeds from all those plants, and then by hand shell them and look at each seed and determine, is there smut fungus on this seed or not? And then count those up with the amount of seeds on that plant, and that becomes a score of how resistant or susceptible that plant is. Now, a field like that I'm showing you actually took 4 months to phenotype with a team of 5 people every day sitting there and doing that. And so this is an extremely laborious process. Again, thinking about plant breeding and how you might breed for a trait like this, doing it in a traditional way is going to be extremely taxing, extremely cumbersome. All right, so we have our population, okay? And now we're taking a step back. We have our population, we have phenotypes. We want to understand the underlying genetics. So if I ask AI, which I did, I said, AI, how might I genotype my population to understand sort of the underlying functional variation that is controlling the trait I'm interested in? There are 3 sort of main genotyping mechanisms at the moment that you might go and work with a company or do yourself, or work with a colleague at your university to help do it for you. So they essentially have 3 different categories, and there's different flavors of them. One that we know very well are DNA microarrays. In my career, I've developed 2 different arrays at the time for peanut. They were incredibly impactful. But one of the things that I was dissatisfied about is that once I designed those markers and it was put on that array, it was fixed. I couldn't change them. And so if I didn't have the markers that really adequately typed and adequately helped me genotype my populations for the traits that I was interested in, it really wasn't as useful as it could have been. But microarrays are accessible. If you have a big community and you're working in a crop that has a lot of people focused on it, they can be relatively cheap. There are actually arrays now at a lower amount of markers that are like pretty cost effective. Another thing that you can do is what's called reduced representation sequencing, which is just sort of cutting up the DNA into different sections and then sequencing those. It's really good for large genomes. It's really good when you don't have a lot of knowledge of the crop that you're working on, or the plant species or animal species that you're working on. The DNA quality doesn't have to be as high as with microarrays or other things, and so there's benefits to that. However, if you look at the name, you're reducing the representation of the genome. You're actually cutting away most of the information that might be impactful and useful for you. And so as usable as that is, it's not giving you all the information that you might have. And the other way to do that is actually sequencing the whole genome. So we've been doing this for a long time in science, and basically, fractionating the genome into small pieces and then sequencing it to a specific depth, usually at a higher depth of coverage, to layer on those reads all across the genome and understand all the variation that exists within that individual. Now, to make it more affordable, you can do what we talked about that's in the title of this webinar, which is actually sequencing that to lower coverage. So if you sequence that to lower coverage, the amount of the cost of the sequencing per sample is reduced, and you can start to sort of think about ramping up the samples that you're going to use. All right, so again, we're going through this naively. We haven't done it before, and we're thinking to ourselves, what are the things that we're going to think about when we try and decide what we're going to do? And so I have a checklist, right? Because with my lab, and probably with your lab or your group or your neck of the woods, you might actually be working for a company. You have a P&L, you have a budget. Everything that you do costs money. It costs you money to come into work. So I think about this a lot too, because through my grants and other sort of revenue sources, I pay my own salary. So when I sit, for instance, when I'm in this webinar, I'm paying myself a certain amount of money per hour. So everything that you do actually costs you money. So when you think about genotyping and how you're going to try and sort of balance the fact that you want to have as much information as possible, your opportunity, right? So you have a population, you spend a lot of resources on phenotyping that. It's very, very important in terms of the global impact. You want to get the best possible information genotypically as you can. The other sort of thing that you're going to think about is how scalable could this be? You might just do it once. It might be a small population, but most of the time what's going to happen is you're going to do it once, it's going to work really well, and you're going to get a taste for it, and you're going to want to do it a lot more because you're going to see how powerful it can be. So how scalable can that be? And then how flexible can it be to tissue type and quality? So I talked to you earlier about all the different tissues and all the different plant and animal species and microbes that we've sequenced and worked with. All of those bring different challenges in terms of DNA, how to get the DNA quality. Really having the ability to have confidence that no matter what you're working with, you're going to have DNA and it's going to go into a process that you're not going to have to redo because it didn't work well. Because every time you have to redo things, that costs you money. And again, in this sort of environment where money becomes a really big limiting step, those situations limit your ability to make the most impact. Okay, so the other thing that's really important that I think about all the time is how are we using our time and resources the most efficient way? Okay, so all of the talented folks that I showed in my slide that are in my lab, every single time they come to work, that costs me money, right? So if I'm wasting their hours because we're putting processes that are sort of not time efficient, we're sort of wasting money that we could be using to help sort of drive other innovations. The resource efficiency is also an important one. So when I was a graduate student, I would just use all the tips I wanted because I could go to another place and there'd be more tips, right? So you think about those things: reagents, beads, tips, plates, tubes. Those things you're just using because they're somewhere in a jar, and when they're out, you ask your boss and they get refilled. But when you have to actually be making the decisions, all of those things are important pieces of the puzzle. And so that time and resource efficiency is also a big one. And then of course, all of that goes into the lowest possible cost. And all of these this sort of checklist, this sort of like ingredient list goes into the topic or the partner of the webinar today, which is Sequel. So Sequel has a product that we use. It's called AgriPrep. And AgriPrep, they say powered by ExpressPlex, which was an older version of this sort of library prep technology. And so I'm going to play a game with my partners at Sequel, and I know they're listening and to this webinar and watching it. And I'm going to first tell them what I think is great about the product, and then I'm going to show you their actual slide where they're telling you what they think is great about the product, and we can see sort of how well it matches up. So the first thing is that this is an incredibly simple process. When I was a graduate student, I was doing RNA-seq about 15 years ago. There was a 2-day process. We made all the reagents ourselves. It felt like it was the most laborious thing I could have imagined. And if I screwed it up, then I'd be fired, right? So those types of processes are extremely cumbersome for folks. This one is not like that. Okay, you have your sort of index plate, which has the indexes and reagents. You have, of course, your DNA. You just mix those two together. That's step one. That's an incredibly simple thing to have to do. It's an amazing, amazing sort of a setup. Then, of course, you do PCR. You know what's great about PCR? You don't have to do anything. You put it in the machine and the PCR does it for you. At that time, which I really like multitasking, you can do other things and you start to sort of layer those things on and you can really scale up your activities in the lab. And then the best part for me is one bead cleanup. So other sort of library preps, you might have 6, 7, 8 times that you would do bead cleanups. Every time you do that, it takes time and energy and focus of an individual that's talented, that you need to be doing important things. Also, those beads cost money. So if we take a step back and look back at this, if I have 10 plates of 96 samples, so 960 individuals, all I have to do is make one sort of pipetting into a reaction plate, do a PCR, and then one bead cleanup for each. That's an incredible process. So we really, really like the way that the AgriPrep is set up. So let's hear from Sequel about what they think is good about their workflow. Okay, so they say it's ultra fast, one-step library prep method. I agree, I just told you that. So they also celebrate that they have pre-plated ready reaction plates. Okay, it's a one-step tagmentation amplification. We're going to talk about auto-normalization, which is a major part of it as well, and it's really fast. They say 100-minute workflow, but that actually includes the PCR. And so if you're thinking to yourself about how you might plan out your day and the things that you might do, this is one of the more sort of exciting sort of preps that you can imagine putting into your workflow to get the results that you want to get. So let's dive into this a little more, because I have an incredibly talented sequencing group lead, Kelly Good, who was a PhD from the University of Georgia. And I asked her to sort of do an analysis of sort of how it looks in terms of the different resources that we use for AgriPrep versus other library prep methods. The first thing that stood out to me was when Kelly sort of calculated how much time it took her to do to prep the samples. So in her sort of estimation, it's 1.5 hours of her time to prep 576 samples. And for me, that's great, because I know that for 6 plates of prep, I'm only paying Kelly for 1.5 hours. And if that's all I need for that day, which normally that's not true, but if it is, she can do other things like upskilling and learning new things and implementing new technologies, because innovation in the lab is so important. So this other part, because we all love dolphins a lot. I love dolphins. One time I swam with dolphins. The dolphin threw me into the fence that we were in, because I think that it didn't like that I was heavy. And so it kind of struggled, and then it was mad, and then it got really aggressive, and then it threw me into the fence. But I still love the dolphins. And so one of the things that we always feel guilt about in the lab is the amount of plastics that we use. So there's 2 ways that I'm sort of talking about this. One is sort of either pipetting it by hand or using automation. And in both of them, this library prep has a massive reduction. So in terms of 20 microliter tips, it's a 67% reduction and almost 100% reduction in 200 microliter tips. Really sort of the workflow is set up to reduce the amount of plastics that you use. And the same sort of reduction you see sort of using automation. So resource and time efficient. So it's checking those boxes. And one of my favorite topics is auto-normalization. I think when I first started doing large scale sort of skim sequencing, I was using library preps that didn't auto-normalize, and we were spending probably 5 to 6 times the amount of time actually quantifying and normalizing plates than we were on the DNA extraction and the prep. But now with sort of products like AgriPrep and having the ability to do the auto-normalization, we only spot check DNA to make sure that the extraction went well. We actually don't do any sort of quantification and normalization. And what I'm showing you is 2 different categories of actual runs. And these could be anything from, again, sort of shed skin DNA to clippings of seeds to leaf tissue to blood or microbiome from the soil or other sources. And at the top is 3 different runs with like a low coverage skim. So we were trying to get around 0.5x to 1x coverage. And again, so we're not doing any quantification. We're going straight into the library prep and sequencing, and we're essentially getting most of the samples well covered in a nice sort of tight CV window, right around 1x coverage. It's really, really nice. And then the bottom is more sort of a higher coverage look. So maybe we're sequencing a little bit more, so 2 to 5x in that case. And again, what you're looking for when you're trying to do these things at scale and you're trying to do them once and not have to do it again is that you're minimizing the attrition, right? Like you're maximizing that it's a one-stop shop in and out. And the auto normalization for agri-prep is really, really good. And when you combine that with all the resource and time efficiencies, it's really sort of an incredible impact on your ability to do the work you want to do. I wanted to talk about automation a little bit. Again, those of you that are really good at automation, which is not me, might say, why would you automate this prep? It's so easy. Well, actually, because it's so easy, automating it actually is a pretty good benefit. So we use the OpenTrons Flex. It's a really simple program. Of course, you just have the DNA and the index in there, and then it pipettes it out. And then after the PCR, it uses one tip to combine all the libraries and do the one bead cleanup. So really incredible and fast and flexible for automation. The other thing that's really great is that SeqWell is willing to sort of make plates that work for your system. So we actually order index reagent plates that have a certain amount of samples for us, so we can have our DNA plates all on the deck, and then just that one reaction plate, and then it does it all sort of in a row. So very, very amenable to automation, even though it's so simple. Okay, so we go back to our wish list, our grocery list. We're doing whole genome sequencing here, so we're extracting the most important information. That's in our opportunity. We have the ability to make it scalable. It's efficient. It's amenable to automation. It's very flexible to tissue type and quality. I didn't really go into that because I think DNA extraction is a little bit boring, although it's the most important aspect of all of this. But we sort of are working with all kinds of different tissues from all kinds of different organisms. It's extremely time and resource efficient, and it hits all those boxes with the cost, because when you calculate cost, all of those reagents, all of that time has to be included in that. And so you're sort of reducing the amount that you have to spend per sample, which allows you to do more work, to sequence more individuals, and to make more of an impact. Okay, if we go back to our example, our global crisis for peanut, our smut. So we had our population, we phenotyped it. Okay, we took a pause and we asked ourselves, how might we genotype it? Well, I'll fast forward to the future. We actually sequenced all of those individuals at the F2 stage and did some mapping. Lucky for us, and again, it doesn't always work out this way. It works out this way a lot, but lucky for us, the resistance resided in an area of the genome we can map very effectively. Because we had this incredible process that was efficient, and we could think about sort of sequencing a lot of individuals, we actually were able to validate it also on a second population, and show very clearly that that's where the resistance lied. So in this case, we have a single reference genome. We mapped it to this location on chromosome 12. We did sort of some really interesting sort of hypothesis testing to decide on some SNPs that we thought were important, and we looked at those in a global sort of broader peanut diversity set that had been phenotyped for this trait, right? And so we asked ourselves the question, if I just selected for this particular allele, this SNP, could I, in a unstructured set of material that aren't related, select for resistance? And what was exciting is that we absolutely could do that. And so we knew for a fact that this was strong resistance with a major gene, and it was in this locus that we mapped. Now, we didn't know exactly what the haplotype was or what the specific gene was, but we knew that this SNP was so tightly linked that we could select for it in a diversity set. And these are two sets of peanut diversity from core collections in the United States and Argentina that have been phenotyped for multiple years for resistance. So, spoiler alert, how might we actually get more information? And that's where sort of the pangenomes come in. Now, if you've worked with pangenomes or seen presentations about pangenomes or thought about them in any way, you know that there's not a great way to sort of show them on a slide. And you can use those sort of like subway tube maps or some of the other sort of graph-based illustrations. They kind of just seem like a big mess. So to introduce pangenomes to you today, what I wanted to do was like look at it in two different ways. In one way, to sort of drive home the point that structural variation has been shown in crops to be extremely impactful drivers of vigor and traits of interest. And so, you know, not being able to actually identify those structural variants and use them in genotyping programs actually limits your ability to find the most useful variation. And on the left, what I'm showing you is an example of that in peanut that I won't go into, but it's basically an extremely strong disease resistance that folks in the industry were trying to map for about 2 decades. And until we were able to use a pangenome approach, we couldn't understand what was the underlying functional variation that controlled that resistance. And as soon as we were able to use a pangenome, it popped right out, and it ended up being this 64 kilobase sort of tandem repeat that was completely invisible before that. So when you think about sort of the power of the pangenome, it's about the ability for you to see more. It's essentially, when I worked in conservation, I was always trying to get better binoculars, right? So I could see we would get up at 5 in the morning and be really quiet and listen for songbirds and try and understand how many species were out there. And so you really wanted to get the best binoculars so you could see those songbirds from far away, because the best ones, as soon as you got close, always flew away. So I think of it like that, is you're continually increasing your resolution of what you can see. The other part about pangenomes that's really impactful is that if you think about it, when you are sequencing a population, okay, you don't know the variation that's in there until you use your genotyping method. And every genotyping method, no matter how good it is relatively incomplete. But when you have the pangenome, you actually have access to all the variation. So my friend Justin said yesterday, you have an incredible set of variants from the beginning, which really, as you'll see later on in the presentation, plays an impactful role in your ability to do the things that you want to do. And one of the things that is abundantly clear about pangenomes and sort of the era that we're in, where we're producing a lot of sort of community-based pangenomes, and we're sort of trying to capture all of the important diversity that we can, limited by money and how many sort of genomes that we can assemble with the people that we have. And it's clear that local pangenomes are very, very important. What I mean by that is when you have a trait of interest or a population of interest, that you have a trait that that is the trait that you want to be able to understand, you have to have the right genomes in your pangenome to actually identify that variation, because it's a closed set of variation. So in this situation, I put cute cats, everyone loves cats, on the screen, and I'm interested in that cat in the middle. So there's a trait that cat in the middle has that I really, really like. And if I have a pangenome that just has these 4 cats represented, there is no way that I'm going to be able to understand the underlying genetics of that really wonderful, it's probably a Russian blue, but it doesn't have the blue eyes, cat in the middle there. So you have to think about that. And I think it makes a lot of sense, but we still have a lot of discussion with folks that we work with about sort of using community pangenomes. And the question is what you're interested in represented in that resource? And the ability to sort of produce these sort of sequences at a lower cost, and it's more accessible, you can start to craft your program around having the most appropriate tools for what you're trying to do. And let me show you what that looks like. So if we go back to our SMUT example, the last time I left you, we had a really great sort of SNP marker that we could select for, and that's wonderful. But if you think about it, we are trying to do breeding in multiple programs across the world, and we don't always want a phenotype, because I talked to you about how cumbersome and labor intensive it was to phenotype for smut resistance. So we really want to be sure what is the functional variation so it's never unlinked in our populations. And using a pangenome, which on the rows there are represented susceptible and resistant peanuts, where you can see very clearly I've indicated there. In that region, there's a clear haplotype that indicates the resistance for smut. And the interesting part about this locus is that that is the largest set of R genes in the entire peanut genome. And so all of these variants that we're showing there, indicated by the dots, are actually variants that are around sort of R genes. And it's going to be really difficult for us to understand which gene is the gene that's controlling that. And so we actually have this resistance haplotype that we can select for. And that becomes extremely powerful. And it's been shown to clearly be the haplotype that is defining resistance to that trait. And we could only do that with a pangenome. We can't do that otherwise. Even if we had one resistant genome and one susceptible, we wouldn't be able to understand the actual haplotype that is sort of defining that resistance. So really powerful. And when we think about then, I have the resistance, but now I'm trying to actually accelerate crop improvement, which is what is in the title of this webinar. Breeding populations represent many founders, okay? And those founders contribute variable genetic backgrounds. And those genetic backgrounds have contributions that are important. And so it's not enough really to just say, I have a marker for this resistance and I can select for that in my breeding program. What I'd like to do is I'd like to select for all kinds of really important traits that are potentially in my breeding populations. And how might I do that effectively? And if we go back and think about this superpower that we've talked about, of this incredibly flexible, time efficient, resource efficient, cost efficient ability to do whole genome sequencing, you can start to craft an idea of how you might use that ability to make incredibly impactful selections in your program. So I've listed a bunch of peanut diseases, and also, of course, you might want to select for seed traits as well. That was just kind of a throw in, but essentially, you might say, okay, these are a bunch of resistant or diseases of peanut, but actually in the populations that we work on, we have known resistance that is segregating for all of these diseases. So we could, in theory, if we got very, very lucky, have peanuts that represent 3, 4, or 5 different resistances. So how do we actually select for that effectively? And that's one of the things that we've been with my colleague Justin Vaughn and Wally Karani have really thought about how we do that effectively for the past couple of years. And that where it comes back to pangenomes. So imagine that your breeding program, and in this case we do, so in our breeding program, we have genomes of all of the founders of our population. And so what I'm showing you is an illustration of how we use mapping to the pangenome to infer haplotypes of individuals using SkimSeq. And so the top one is 3 different chromosomes. They're colored by founder contribution. So let's just look at the left sort of set of graphs there. So the top one is mom, and mom is a hybrid. And you can see mom as haplotype orange and haplotype green. And the second one down there is dad. And there's a little bit of noise in there because, again, we're looking at SkimSeq, but we're inferring sort of haplotypes using the pangenome graph. Dad is haplotype pink and haplotype light orange. And then the one of the progeny of that cross, we've named Kid, which is a very sort of appropriate name because that's what I call my kids, Kid 1 and Kid 2. So you can see very clearly the haplotypes, one from dad, one from mom, and then where recombination happened. And so imagine, and so this illustration, you think about in your head, okay, I have an entire genome. I have the ability to essentially paint founder haplotype contribution across the entire genome. And you can start to make very clever selections about what haplotypes you want to have as a sort of superior sort of genotype that you're maybe selecting for. And not just 1 or 2 traits, but traits across the entire genome. An incredibly powerful sort of superpower to have. And this sort of table is basically one of the sets of traits that we actually select for in our program. So we're selecting for the fatty acid composition of the seed. We're selecting for resistance to smut. We're selecting for resistance to leaf spot and virus and stem rot, sclerotinia blight. We're selecting for lower aflatoxin contamination. We actually have QTLs that we can select for drought tolerance that have been validated across different populations. And we can very clearly and cleanly infer sort of the contributions of the different parents in those progenies and say, this individual has a contribution at this site from this parent, and understand sort of what phenotypes those sort of progeny are going to have. And again, if I step back to the whole sort of premise and the whole theme of this presentation and webinar, if you look and think in your mind very clearly about your program and the traits that are of interest to you and what can be impactful and what you're trying to discover, really, I can imagine you might be getting excited about the possibilities. And I don't want to just talk about, the role of SkimSeq and pangenomes in my own program. I'd like to sort of show some published examples that are exciting. And here's an example from Sarah Carey, who's here at HudsonAlpha, a really talented scientist, where she used sequencing a very large population, backcrop cross-population of hemp and a pangenome that she produced to understand sort of the sex determination in cannabis, and then actually was able to link that up to the sex determination in hop as well. That paper is wonderful, and if you sort of want to see a different example of how this can work, that's an excellent example. Another example from Ethan Thompson, who's a postdoc in my lab, who got his PhD at the University of Georgia, is something that we're very excited about because it has an extremely important impact on the industry of peanut worldwide. So there's a dichotomy between what peanut butter peanuts want and what confectionery peanuts want, and most of the world wants peanut that's high oleic acid, and the peanut butter folks want a peanut that's mid-oleic or what we call normal oleic acid. And so for a long time, there was potentially two genes, fatty acid desaturase genes, that the industry thought controlled sort of that fatty acid composition. And no other sets of markers popped up in any sort of mapping. It was always those two genes. Well, Ethan, working with a pangenome of a population, used sort of fatty acid composition as a control, and thinking that if this experiment works the way I expect, I will get these two genes that will pop up, and I understand that my population is good. Well, what he noticed is that there was actually signal a little bit far away from the one gene, and it turned out that there's a third gene. And in this paper, he showcases how we show that this gene is actually important in driving sort of the different ratios of oleic to linoleic acid, which is going to have a big impact on the industry and how they select for these different peanut varieties in terms of exports, imports, and who they're selling them to. So another really interesting example of how pangenomes and skim sequencing unlocked sort of a mystery that probably wouldn't have been seen otherwise. And then one more example, and this one's kind of like not as interesting and a little more sort of generic, but this is a group that we worked with where we were mapping a trait for them that's really impactful for their sort of program. And the way that we do mapping, in some cases, we do something called QTL sequencing, where we sequence the bulks representing of a population that represent the tails of the phenotypic distribution. And then we look at genome-wide allele frequencies, and you can find a peak where sort of the phenotype is being controlled by the underlying genotype. And on the left, when we did this just on a single reference genome that didn't have the trait of interest, we saw the signal and the peak, which I'm highlighting there, but it was kind of noisy and it'd be difficult to say where it might be on the chromosome. But when we used a specific local pangenome graph approach, on the right It's a very, very clear peak in sort of the right position that we are expecting, and very clear sort of underlying variation in genes that make a lot of sense that are now being followed up on. And so those are sort of two situations where if you didn't have the pangenome, you probably would have done the mapping and then done a lot of follow-up and time and energy and cost and not really understanding the underlying functional variation, whereas having that pangenome, the same experiment allowed you to get to that quickly. And so imagine that in your mind in terms of time and cost and opportunity. And it's a major, major impact. And again, back to our example. So there's me in front of a peanut variety that is smut resistant, it's high oleic, and it has other disease resistances as well. And from the time that we identified the smut resistance to the time where we were able to develop that peanut variety was right around 3 years. And so starting from the beginning and looking at all of the sort of steps that I walked you through of having the ability to sequence the populations at scale quickly, to interpret them on a pangenome graph, to understand the most important variation, and then to do that selection, gave us this superpower to be able to take this global crisis and produce a tool that across multiple breeding programs that is going to be impactful for those growers and the industry moving forward. And stepping back and looking at sort of the bottom line here is that really we're at a point where skim sequencing and whole genome sequencing combined with sort of specific and local pangenome graphs are incredibly powerful tools for gene discovery and selection. And no matter what you're focused on and what you're trying to do, you always have to understand what gene you might edit, what gene you might select for, what variant is important in your populations for the traits of interest. And so the ability to do this in such a simple way and cost-effective way, I think is really incredible and exciting. And there are products out there that can help you do that, but Sequel AgriPrep is really a wonderful product. It's scalable, it's cost-effective, and it's resource efficient, and produces superior data. And I would be remiss if I didn't talk to you about sort of the program here at Hudson Alpha, because, hey, I understand. You might say, that's wonderful. I don't have the resources or the time or the people to do those things, but I still want to be able to do it. And we're here to help you. So if you're sort of optimizing your selection, you're scaling genotyping, or you're trying to understand sort of gene identification and discovery, we're here to help. We have done this a lot over the past 5 years in many different systems, and we're happy to sort of help you make the impact that you're trying to make in your neck of the woods. And with that, with acknowledgments, I have to acknowledge for the SMUT story, the leader of that really is Kelly Chamberlain, who works for the USDA, and that's in collaboration with Mars Snacking, led by Peggy Sastos, and also in collaboration with INTA in Argentina. Jorge Baldassari is the breeder there, incredibly talented breeding. So Kelly and Jorge and Peggy, and then Corley Holbrook at the USDA as well, are really the main players that we're working with to sort of identify that resistance and then do the breeding. And on the Hudson Alpha side, a lot of the stuff that you did, I talked about Waleed and Justin, but a lot of the stuff that you saw really is done by our computational team, Hallie, Zach Myers, and Catherine Davis. And of course, our sequencing lead, Kelly Good, and a really talented research associate, Ambrielle White, who helps with the DNA extraction. And with that, I'm happy to take any questions that you may have. Thank you.
Great. Thank you, Josh. And thank you again to the audience for joining us today. We will now start the question and answer portion. Please submit your questions. If you hear a question that we're talking about and you want to ask more about that, please do. We want to make this as interactive as possible. So I will get on here and start asking. Josh, are you ready?
I'm ready.
All right. Fabio asked, what was the most difficult step that you had to adapt to introduce the LP whole genome sequencing in the routine?
Yeah, that's a awesome question, actually. So all the steps are difficult, but I think the most difficult step is really dialing in the DNA extraction, um, because if you start, so if you're extracting DNA in plates from small amounts of tissue, especially from plants where you have lots of different compounds that might make the downstream um, uh, activities more difficult. Um, that's a really hard step, and it's one of the things that actually we spend the most time on dialing in, which is also another awesome sort of tip of the cap to the library prep, is that like that's kind of the afterthought. Um, we use, I'll just say it, we use LGC SBDX DNA extraction. We like that a lot. That's been the most difficult part. Now, the analysis part is very complex. One of the first thing that we did was we developed the analytics. And, it's one thing to sequence thousands of samples. It's another thing to sort of get them aligned, have the variation called and imputed, and then interpreted on the back end. So that's another piece that becomes difficult. Although there's lots of really good tools that'll help you there, and of course we also provide that service if somebody needs that. But that's the first thing that we put in place, and that's a pretty complicated part of the process.
Great, thank you. All right, David, how do you deal with heterogeneity within any given variety in your diversity panel? Do you sequence multiple individuals from each variety?
Yeah, it's a great question, and it comes up a lot. So with some of the different folks that we work with, so um, we've done multiple things. So it really depends on what you're trying to do. I think if I use an example from maybe national plant germplasm folks, where they do have heterogeneous sort of accessions, and they're kind of interested in allele frequencies within those accessions and how much diversity is within them. We do sequence multiple individuals. What we'll do is we'll basically extract DNA from multiple seeds in that well, and then we get that indication. Now, with skim sequencing, there's a little bit of bias, and so you can get a little bit higher depth of your loci in the first pass. But normally, when we do that for folks, and that's an important aspect of what they're trying to do, we use a little bit different approach where we do an enrichment capture panel that essentially targets about 100,000 different sites, and then that gets higher depth of coverage. What's really cool about that is that you can do that on top of your whole genome sequencing and get it at the same time. And because sequencing technology across the different providers has become so cost effective, that becomes sort of an important tool there as well. But yes, we will either, if that's important, we will sequence multiple individuals. If instead you just want to get a representative, we, in some cases, would just sequence one. But that's how we do that.
Great. Carolina, QTL sequencing was skim-seq, ?
Yeah, I didn't go over that. So that's a good question. It's one of my favorite things about sort of what we do. So when I was doing my PhD and my postdoc, we were doing QTL-seq with bulks, and you would essentially either extract the DNA from all your individuals and pool it and try and like get, you want to get equal representation from each. Or one of the times what we did is we just took leaf punches of all the individuals into the extraction and did that extraction and then sequenced that. And that's cool and it works really well. But like what's really interesting about sort of QTL-seq is that the phenotypic error gets in the way, right? So you have some where you think they're resistant or you think they're susceptible, but it's really an escape or a false positive or negative. And once you sequence that bulk, like you're kind of locked in. So when we started having the ability to do sort of large scale skim sequencing, I really like this idea of doing QTL-seq on populations, but actually sequencing each individual. And so we do the bulking in silico. So the sequencing is skim-seq, and then those are brought together, and the bulking is in silico. What's also beneficial about that is it allows you to do fine mapping if you want. It allows you to separate potential false positives or negatives and take them out. And we actually do some procedures where we sort of run it iteratively and find the ones that are not really behaving and get rid of those. And then it also lets you do sort of smaller experiments. So in my life, for my sort of personal sort of work, we always sequence the whole population. But for some folks, what we do, in fact, there's a really great watermelon group at University of Georgia that we work with a lot. And what they do is they just send us the bulks and individuals. So we just do one plate for them, and then we do the bulking in silico and do the mapping that way. So there's a lot more power involved. But yes, you're right. It's skim-seq on each individual, and then those are sort of bulked together on the computer.
Great. And that leads right into this next question, which I don't think you got into the long reads of it, but Raja says, great talk, thanks. Whether reduced representation skim-seq will give long reads even after digestion.
Yeah, that's a kind of convoluted question. So on the long read side, we don't generally prep sort of the long read libraries from DNA preps that we're using on the large scale genotyping portion. Yeah. So once you sort of digest DNA, you're kind of left with the fragment size that you have. So that's a sort of a separate sort of entity in itself. So we don't in fact do the long read prep and sequencing from the same samples. But I will say, and I can do this plug, Sequel has a product called Longplex, which actually uses similar tagmentation technology to do scaled and multiplex long read sequencing. But the DNA going in needs to be extracted in a high molecular weight fashion before you use that. So those are two different things.
Great. And this could be a long answer on this question. We may want to follow up with an email, but I'll start it here. Fifi says, how was the auto normalization done? Kindly explain how that works with the AgriPrep.
Yes. So actually, I could definitely answer that, but Sequel has great documentation of how that works on their website, and I don't want to screw it up. So yeah, I think that's like very nicely documented in their materials. But I can tell you that it works wonderfully, and it has to do with sort of the amount of reagents and the rate limiting step of the tagmentation that sort of takes whatever sort of DNA you have, and the library that is prepped from that process ends up being similar, and then you can pool those effectively. So that's the general idea. But if you want more sort of nuts and bolts, they have really good documentation about that.
And we'll make sure to follow up with you on that question with more information, a link to where that sits, all that good information there via email. So thank you for the question, though. We appreciate it. All right, here we go. Wonderful presentation. This is a multi-question kind of question here that was typed in. Again, wonderful presentation. What marker-based system you have used to screen the mapping population once after your computational discoveries?
Yeah, great question. So at this point in time, we mostly just use skim-seq on everything because we've got it to the point that in my hands it's a certain cost, which is extremely low, which I won't share, but it's effective for us just to do that. In some cases, we will use sort of TACMan for individual markers to validate if we want to do that and just make sure. What I didn't go into today is the ability to use enrichment panels, which I'd mentioned before. And so most of our sort of breeding operation and the folks that we work with where we're doing large scale sort of selections for them, we use sort of custom enrichment panels, which you can order either from Twist Bioscience or from IDT or others that would produce oligos. And it's based on, there's biotin on those probes, and those probes sort of, you introduce them to the library, and you do an incubation, and basically then use biotin sort of beads to take all those enriched portions and connect them to the beads, and then wash everything away. And then what you're left with is just that sort of set of molecules that you're interested in. That works for a really good sort of targeted marker system. And again, what we do most of the time is we get those sort of enriched portion, and we bring the whole genome sequencing library back in. And so that's mostly like how we do it, instead of like, you know, doing individual marker assays. Because again, when I go back to sort of the main point is that I want to have the most important information on every sample, right? So if I run an individual marker on the sample, I just have that one marker. But if I do everything on that sample, I can use that information for all sorts of different things. So that's generally what we do. But yeah, I mean, you can use sort of TACMan assays, and actually we did that for something recently, and I was pretty impressed. About 10 years ago, when I used to do sort of CASP, we would sort of make our own, but now they do it for you. It's all very nice, and it works really well. So that's how we do it.
Here's a second part of that question, and I know it's 9 o'clock, 9:02, so we're, well, on the West Coast anyway. We're at that time for the webinar, but we still have almost 200 people on. So do you mind if we follow up on a couple more questions? Is that okay with you, Josh?
I don't mind.
All right, let's do it. We'll stay on for a few. We have 200 people on, so we might as well. So this is the second part of that question. Can we develop a mapping population from founders, not from pangenome panel, and identify the variants by pangenome-wide association studies?
Yes, exactly. Absolutely. In fact,
yeah.
that's exactly what we do. So I didn't go into the genome construction and the long read stuff, because the webinar is about SQL, so I wanted to focus on those things. But you know, I'm a PacBio shop. I run a Revio. Hi-Fi sequencing with the new SparkNext technology is incredibly cost effective. We can do a peanut genome for less than $1,000, which is incredible, because the first one that I worked on, 15 years ago, cost $6 million. And so now, we can do one for less than $1,000. And a peanut genome is about 2.5 gigabases. So whatever system you're working on, whether it's less than that or more than that, you can create your pangenome based on the founders in your sort of neck of the woods. And as I sort of showcased in our work, that becomes amazingly impactful. And when we have other projects with other breeding programs and groups where we're producing sort of pangenomes for just their sort of populations that are important for the work that they're doing. And the other thing that I really like about that, and what we do in our breeding program, is that every time we introduce new parents, we just sequence that genome, and now it becomes part of our sort of overall pangenome. And as we sort of produce that, it's really just this living tool that's really important and impactful. So the ways that you might do that effectively, of course, work with a partner like my lab and my colleague Alex Harkis. We run the Revio, and we do a great job of we have a person, Paige Phelps, who is part of the lab, who is really wonderful at getting high molecular weight DNA and prepping those for sequencing to produce those graphs. And so we very routinely do this for folks, where we actually are producing pangenome graphs for them, and then doing the population sequencing and gene discovery interpretation on top of that. So if you wanted to do that in your own program, there's lots of sort of ways that you can have access to that, and it's very accessible now. And I highly encourage that you do that, because I think I enjoy sort of the community-based aspect of community pangenomes, but like, if you are working in a situation where the traits that you're interested in aren't represented in those individuals, they're not going to be useful for you. It's more of a sort of basic science sort of type of thing. But if you were trying to really do that applied and translational work, you have to have the variation in your hands to be able to identify it and utilize it. So yeah, good question. Thank you. These are all really good. Did you sort of produce these questions, Drew, or were these actual questions that people are asking?
I just keep coming up with them as you talk as well, because here's another good one that fits right into that. I'm glad you're talking about graphing there and doing this, because Joshua said, and you kind of answered this part way, but I'll let you elaborate on it a little more. How might ascertainment bias in relation to developing pangenome graphs pose issues, especially when considering interspecies comparisons?
Absolutely. Yeah. I mean, so here's where we're getting into the nerdy stuff, which I don't get to like think about enough, but it's an important consideration based on what you're working on. And so certainly again, like ascertainment bias is something that I've thought a lot about, and with sort of developing SNP arrays. And actually in Peanut, we developed the array in a way that had markers that we thought could type different wild species, because there's great programs that sort of introduce sort of wild species segments into cultivated peanut to develop sort of new traits in that way. And it becomes really difficult. And if you have sort of a situation where you have different sort of species that you're crossing together, where the graphs become a little more complex, you certainly have to have all of the individuals that are going to be contributing sort of haplotypes in your graph to not have those sort of ascertainment biases in your downstream analysis. Now, something that is kind of like what we're working on now, which I'm really excited about, that Justin Vaughn is working on, is actually trying to understand sort of incomplete pangenome graphs and inferring haplotypes of founders that you don't necessarily have in your graph, and how might that work. And we're thinking about sort of how to do that. But if you imagine, this is something that I didn't really explain as well as I could have in the webinar, but it's really kind of embedded within the name. It's a pangenome graph, right? So if you think about a graph abstractly, the things that are on the graph are the things that are on the graph. Like there's nothing there that's not like that you can't see. And the pangenome graph is the same way. So if I use 3 genomes, like the only thing I have access to using that graph is the variation in those genomes. So anything else that comes on or that is part of my population, I don't have access to, and I can't see. And so, yeah, you end up getting that sort of ascertainment bias. So that's why this sort of specific creation of graphs that fit your purpose is really important. And thinking through sort of that process, and it's worth it. It might feel like, oh, I would have to do 10 genomes to do this, but trust me, it's worth it. I do this so much, and the things that we're able to find and see and use, either gene discovery or selection, without that initial investment, which is less and less and less, and way cheaper than it was before, we wouldn't have been able to do. And so, yeah, so you have to do that in a way, in my opinion, but also we're at a place where it's extremely accessible and cost effective. And then just this amazing sort of, I always think back to, I'm old, I'm not super old, but I'm like mid-old. And when the Genesis came out, some of you may know what that is. I think I was like 6 or 7, and we wanted the Genesis for Christmas. And I came down the stairs and my brother was yelling, and like I was so excited I went blind, and I like couldn't see like the present. And I always think back to that moment when I'm thinking about sort of what you can do with a pangenome graph that's specific to your part of the world, and then layering on this incredibly like accessible library prep that you can do skim sequencing of thousands and thousands of individuals, and then use that in your genetics. I think it's a really exciting. It's about, I'm not blind right now, but I'm about excited as I was when I found out I was going to get that Genesis and I could play Altered Beast and lose after the second round. But anyway, that's probably answering that question.
Thank you. I love this interactive stuff here. This is great. Okay, so Helen, we're going to keep going here for a minute. We still have over 150 on, so let's get through. We have only a couple more questions. Helen, thank you for this great presentation. You said that the diversity you put in your pangenome is critical for the downstream analysis. What do you think of pangenome augmentation? Oh, I just lost it. Oh, sorry. The pangenome augmentation with the low pass in case of diversity analysis, for example.
Yeah, absolutely. I think that's a great question and prompt, and that's something that we're working on. I also have a company that I founded called Veil Genomics, and we think about that a lot because Veil Genomics specializes in long read, low pass sequencing, which that's sort of a way to sort of do that graph augmentation with structural variation and SNP variation using sort of lower coverage long read sequencing of a set of lines that are going to be important that you might not want to invest a full genome in. And I think back to groups that we talk to who are doing hundreds of thousands of individuals a year for their breeding program. And they want to sequence a small amount of sites and then impute the rest for their genomic prediction schemes. And they say, well, I have all my founders and there's 50 founders, right? And so I want to do like 50x short read sequencing on the founders, identify all the important variation, and then I can do that imputation in my breeding program. And that is basically getting to what you're saying, right? So finding a way to sort of augment the graph with sort of high quality variants, which could be low pass either with long reads or short reads, and then creating that sort of new graph. If you use short reads, you can't understand structural variants. If you use long reads, you can. And so there's different trade-offs. But yeah, definitely. We also have a project on finger millet where we have a finger millet diversity set that we've gotten long reads over across the whole set there. And we can actually create the graph from those lower coverage long reads and then be able to utilize that. And that represents like 300 lines that represents the diversity of finger millet. So yeah, you're thinking in the right way. That's a good question.
Great. Ruth, what coverage of the observation do you need to find variance? So what coverage of this do you need to find the variance?
Yeah, right. So we like to target right around 1x. So depending on your system, you might be using Illumina sequencing, you might be using element sequencing, which also has a really great product. You might be using Ultima sequencing, you might be using something else, MGI or something. And you're sort of thinking about sort of the trade-off between what you have and how much you paid. So for us right now, the sweet spot, we use Illumina. The sweet spot is right around 1x of most genomes. Once you get to a certain sort of genome size, we go lower and you look at half x and quarter x. You sort of, in your large populations, you sort of have more missing data at the lower coverage, but depending on your system, you can impute pretty easily. And so there's that trade-off there. But yeah, I think the vast majority of the time with my personal projects, I'm targeting around 1x coverage.
Thank you. Okay, last question. I promise. I don't know if it's you or me, but people are starting to drop off now, maybe due to the time. I'm going to blame it on it was great. So it's probably my fault they're dropping off. But Jay, here he goes. Fantastic presentation. Thank you. You mentioned a bit about painting the pangenome with founder haplotypes. Do you think there is any utility in using this to integrate QTL results from past linkage GWAS-based analysis by being able to more confidently see if a progeny has an identical segment from a parent of one of those past populations where the QTL was found if that parent is in the pangenome. Woo!
Let me answer that question.
To end it off.
Yes. Yes. Absolutely.
Yeah.
Yes.
There we go.
So that's what excites us, right? So like you have those haplotypes, and so you can do any sort of querying you want. You can look at putting in all the QTLs, and I know that Justin has something called GeneSieve that he's produced that does something similar. You can look at putting all the QTLs from past studies into sort of a model, and then taking your haplotypes and putting them into the model and saying what QTLs might I have that I didn't know I had. Yes, all of that. And that's sort of like the exciting part about this is like once you can effectively infer those haplotypes at scale, there's all kinds of information you can layer on in different ways depending on what you're trying to do. So great, sort of, you're thinking exactly the right way, and you absolutely could and should do that, and we should do that. And whoever is going to make that, I'd be excited to use it in my own program, because that's exactly sort of the type of things that we can do. So that's an excellent question to end it, Drew, I think.
There we go. I love it. Well, Josh, thank you so much. Again, that presentation was great. We really thank you. Thank you, Sequel, for supporting this PAG webinar series. Thank you for Sequel for making this webinar happen for all these people. It's amazing information. Thank you all for joining us today. Look for the post-webinar survey via email. It's very important. Please fill that out. Your feedback is important to us. The on-demand webinar will also be available on the PAG website, so you can, if you missed anything and want to go back, you can always look at that or send it to somebody who you think might be interested in it. We look forward to your attending future PAG webinars, and we will see you in January at PAG 34. So thank you, everyone.